Hello, we are going to discuss our second case study, which is grasping biological cells. Here is where compliant mechanisms become very useful to have a very distinct technique for handling single biological cells. So, let us look at this second case study where compliant mechanisms go down to the micro scale and are able to manipulate single biological cells and also try to measure the forces being acted upon them. This work was done with my former students Santosh Bhargav, Annam Narayan Reddy and Deepak Sahu, who was the one first made the gripper in our lab to hold biological cells, and the two PhD students Santosh and Reddy did a lot, and there are a number of other students who contributed to this work, and in fact this list may not be exhaustive. So, I always start with this microscope image to say that it was Robert Hooke, one of the pioneers in two fields, cell biology and mechanics of solids, because the Hooke's law that we talk about today is credited to Robert Hooke, who said, using an anagram in Latin, attentio sequis, which means the force varies as the stretch. That is what he had observed. He also had observed the cork cells under the microscope and realized that living organisms are made of building blocks which are cells. As we know, biological cells, they are of the micron size. So, cell biology and mechanics were linked at their very inception. Okay. It is actually quite interesting today when we work on mechanical engineers work on biological cells or biology. It is not at all surprising, because the very originator of in a way for both fields, cell biology and mechanics, Robert Hooke had done that, one man. It is also very interesting to note that Francis Crick, the Crick and Watson fame, Watson and Crick fame, in 1949 had published a paper, 1950 another paper. So, they are received here and then published there. There are two parts, this is a part 1 and part 2, where talking about physical properties of cytoplasm. So, he is talking about using a mechanical method for probing biological cells. In fact, it is interesting this year 1950-1949, because just a few years before he discovered along with Watson, Jim Watson, the double helix structures. He was interested in mechanics of cells so long ago, and there are papers that go back to 1938 and so forth as well. So, Crick's description of a biological cell is important. As you listen to a mechanical engineer talking about biological cells, he talked about a cell as if it is a mother's work basket. In his time, I guess mothers used to sew things, and there were jumble of beads and buttons of all shapes and sizes, and there are pins, and there are threads and so forth. They are all jostling about inside the cell due to colloidal forces. He himself put it in under quotes. So, basically cell is a complicated thing, and understanding it from mechanical viewpoint is what many people are doing under the name of biomechanics of cell or mechanobiology of the cell. What is of importance to us now is that Crick had devised instruments for twisting, dragging and prodding cells in order to understand. These are taken from his paper, the excerpts. Okay. So, when we say grasping cells, we literally mean that we want to grasp it like a gripper, but what we actually mean is thinking about cells and understanding how cells behave from the viewpoint of mechanical. Both are there in the word grasp. Now, it as a pun is intended. So, there are a lot of things that are needed for this. First, we need this miniature grippers, that is what we will focus on in this lecture, but we also need to sense forces, which also can be done using compliant mechanisms. Compliant mechanisms help in this 1 and also 2. And then with these things we can do 3, we can measure bulk stiffness, we can also measure their interior properties, which is this 4. So, compliant mechanisms play an important role in this particular field. You can manipulate cells in many ways, but there is an advantage with the compliant mechanisms. When you do these grippers, you do not use any other field, meaning that there is no electrical, magnetic. The Crick had used magneto-twisting cytometry as it is called today. So, you do not have any other energy field affecting the cells, because what all things affect a cell is not known. People are still trying to figuring it out. So, if you have a mechanical one, you do not have interference with anything, it is no thermal. If you use laser tweezers, there could be temperature changes in the cell, that might also make cell behave differently. So, here we are using purely mechanically and in the water environment of the cells. So, without going to the details, first I am going to show you a few movies or video clippings of doing all kinds of these manipulations: grasping, squeezing, piercing, rolling, peeling, bending, probing, stretching. So, all the things, just to get an appreciation for this. Again, this lecture, all the case study lectures have a lot more information in the slides than what we will have time to discuss all this. Just let us see what compliant mechanisms can do. First is grasp. What you see here is a compliant mechanism. There are a number of cells here, these are MCF7 cells, these are mammary gland cancer cells. There is a small pipette which is used only to position the cell and does not do anything. There is a dark shade here, that is the probe that is actually going to push this here. This is fixed here as well as there, and this is made of SU8 polymer, and it can hold this single cell, and this is made of SU8, and this dimension is about 5 or 7 microns. The cell itself will be about the same size. So, about 10 microns in size. You can see that in the movie. So, this is just positioning it, and this one is holding it, and you will see when it comes into focus the cell actually being grasped and squeezed a little bit also. So, that is where the cell is. So, this is only to position, and it is able to grasp it and do this as this one input is there. Okay. So, we can do that now with compliant mechanisms. And next thing is squeezing. The squeezing is really squeezing a lot, as you will see, and this is also done with compliant grippers, where you are going to see the jaws. It is actually squeezed it and then let it relax real time. So, it takes a while. So, you can see it when it played again. When you apply force somewhere else, this compliant mechanism kind of squeezes it. So, yes, it should play. Okay, let us do this. I think it had to go back. Yeah, it just squeezes and then it just recovers and stays there. Okay. And you can also pierce and inject things into it. Here we are just calling it piercing because we are not injecting anything into the cell, just to show that we can actually do this. Here is in fact, here it is a zebrafish embryo, but we can also do the single cells. With a pipette we are piercing it, and a compliant mechanism is holding it in place while it is being done. And we can also roll a cell, as will be seen in this next movie. So, you can actually make it roll as if it is between your fingers, and a number of mechanisms were designed recently. Also one I showed in one of the previous lectures. You can grasp and roll using one single or two inputs that are coordinated, and you can peel a cell. This is a particular special type of cell where you can peel them off their substrate, and here you do not see the compliant mechanism, but what you see, what you do not see are these two probes to which a micronewton force sensor was attached, which we will discuss another case study. So, it is more of manipulation capability with the probes, and those probes are attached to a compliant mechanism that measures forces. You can peel one cell which is attached to another one by just tearing them apart. And you can also bend a cell, and most of the cells, bending of a cell does not make a whole lot of sense, but for some cells it does matter. As we see in this video, these are particular type of cells, they are very special type of cells where they are flat and you can actually bend it here. So, you can see this with probes, again probes are attached to the force sensors, which are again compliant mechanisms. So, it is bent now, and you can probe it. So, here you can see this needle that is probing is very flexible, it is bending so much more compared to the cell itself, and all these forces can be measured. And stretching we had already seen. One point to notice here is that the material of this mechanism and the one inside is different, they are different. So, the reason is that if you use lithography and make the micro compliant mechanism, here it is supposed to grasp something and then stretch, you need to have very tiny beams. So, with those tiny beams making something this big is not possible. The process that makes the big beam, a big compliant mechanism cannot make this. So, what we have ended up doing is, in this composite compliant mechanism, so to speak, composite meaning the two of them are together, not it is made of composite materials. So, we have 3D printed this white one, and then this small one is SU8 made, lithographically made and put into that. So, that is how it is done, and this video of this we have seen earlier, how we can use a single input where you can open up things over here, you put a little thing there, it can grab it and also stretch it. So, this video shows that. So, how when these is also a contact aided compliant mechanism, when these things touch, that is when these things open up. It is all one single piece, it opens up. Now, you put something, remove a force, then it will hold it and keep it. After that, when you press it, it is going to actually stretch it, as you can see. So, once it is placed, now when you pull it the other way, this starts stretching. So, things were coming together when you push, when you pull they go apart. So, you can actually grab something and then stretch. The micro version of this is of course, as I said, it has to be made in two parts. Okay. With all of these things, force sensing can also be done with compliant mechanism, not only manipulation, but also force sensing, which we will discuss in another lecture. But what we will focus on here, in fact, this is the force sensor compliant mechanism. This is a compliant that grabs the cell, and this one, whatever little force is there, it will amplify the displacement over there, which you measure using a digital microscope, and we have a force sensor as well. This is a setup in our laboratory. This is a digital microscope that measures the output displacement and gives us the force, while compliant mechanisms are actually manipulating the biological cell. If you look at the design aspect, we looked at a number of grippers, and each gripper has its own characteristic. If you see, in fact, the other day when we looked at our database, there were 24 or so grippers in our collection. All of them have, we saw this gripper just now holding a cancer cell here, a breast cancer cell, and these are lot of other ones. So, this is something that we just saw another movie where this thing come together. In this empty space we put DACM, so we can measure the force as well, and this reducing the stiffness down to the stiffness of the cell, which can vary anywhere from 1 Newton per meter all the way to 1 millinewton per meter. So, it is a very low stiffness that biological cells have. So, if you want to handle them gently without causing damage to them, you have to have this range of stiffness to your mechanisms also. That is why a lot of tricks were played out. One is just optimal design, first you make optimization, topology, shape or whatever, and then composite, you put one inside the other, and you also add, occasionally we had tried to add negative stiffness elements. By that what I mean, I will show you a picture later. So, one inside the other, as we just saw, definitely makes a lot of sense, and we can put an array of them. So, not one, many cells can be grasped at the same time. Negative stiffness is, we have talked about bistable elements. Bistable elements over a small range, they have negative stiffness. If we attach that preloaded along with the gripper, then the gripper overall has some positive stiffness, this has negative stiffness over a small range. You put them in parallel, overall the stiffness would reduce. That is the idea, we will reduce by even order of magnitude. So, to get down to that level. And we also use this non-dimensional maps to understand the capability of geometric advantage, mechanical advantage and the force required, stresses and so forth in doing this. And sometimes we can use these maps to decide the size as we had discussed once. So, what should be the size of this? So, you have the characteristic length L, let us say this one is L from here to here, how much that should be, in this case it turned out to be 640 microns based on your fabrication capability. This is the thickness, B in this case is out of plane thickness, D is in plane width, that is 5 microns. Somebody says with fabrication they can only make 5 micron beams, we can put that. If you want to measure 1 nanonewton force, and from this map you can get this eta and assume in this case it is SU8 materials 4 gigapascal Young's modulus. If you want to have at least 2 microns to see under microscope measuring this displacement, smaller than that if you think it is difficult, 1 nanonewton giving out output displacement 2 microns, it says that size should be 640 microns. So, accordingly you can design, and putting one inside the other we already talked about, where you have one mechanism that gives the parallel jaw motion and within that we put 2 DACMs and this is where the cell is going to go. So, this tiny space is where it is going to go and these beams are only 5 microns wide, okay, and the cell as we can see that is also a few microns. And this one is 10 millimeters by 10 millimeters, this big mechanism, within that is only 1.7 by about 1.7. So, about 2 millimeters thing, and then these things are very small and they do not break. We made sure that stress are small and range of displacement here is only to squeeze by about 10 microns. There is a process that was developed that is also very important, and what material we are using here, using SU8, and there are a lot more difficulties in this. It is not just the designing compliant mechanism. When you try to put it in water, sometimes the buoyancy force makes them go out up, while the cells are sitting there, these things will go up like this. So, we have to play with a lot of parameters even in fabrication to make this. Finally, when we do that, we have the mechanism and this little mechanism over there, that is where the cell would go and grasp. Okay. So, a compliant mechanism can be used to manipulate a biological cell in many ways. So, whatever you think of including tearing, you can actually tear a cell apart with mechanical tools. One can use just beams, but then compliant mechanisms themselves are made up of beam segments. So, we have to arrange them in a way that the manipulation that we are interested is obtained here, and in the next case study we will see how compliant mechanisms can be used to measure force as well. Okay. Thank you.